Okay, let's go ahead and look at our first protein concept map. So if we start at the top left, our proteins are made of a total of 20 amino acids. Of those, we have 9 that are essential. And if you recall, our private Matt Hill is our mnemonic for our 9 essential amino acids. We have some uncommon amino acids as well, some that are found in collagen, and for instance, we have hydroxyproline and hydroxylysine. So hydroxyproline and hydroxylysine. There's one that's found in elastin that's called desmosine. And then one that's used in clotting, which is carboxyglutamate, which is just a glutamate amino acid with an extra carboxylic acid on it. So carboxyglutamate. I have listed out here selenocysteine, which is just a cysteine molecule instead of a sulfur. It has selenium, the element selenium in it, replacing the sulfur. Some of our functions of amino acids are buffering. When it comes to buffering, we have PK1, PK2, and PKR. PK1 is associated with the carboxylic acid. We have our PK2 is associated with our amino group. And then we have our PKRs associated with our R group. The PKR is the most important PK for buffering because there's a lot more PKRs compared to every peptide has a single PK1 and a single PK2 with a whole bunch of PKRs in between. For buffering, we have a net neutral charge when we're at the isoelectric point. Oops. When we have a net zero charge, it's called a Zwitter ion. And that's when we're at the isoelectric point. So that's our line here is at our isoelectric point we have our Zwitter ion, which is where we are the worst buffer by far at the isoelectric point. We're much more likely to precipitate out under that solution. Your best buffer, you're going to be your best buffer right around the pKs. So your good buffers, remember those plateau points where your good buffers around the pKs, that point where we have a big spike, and pH is where we have our isoelectric point. So for our proteins then, we can make our amino acids into peptides. Our peptide bonds are formed by dehydration, where those are all dehydration or condensation reactions. With our peptides and proteins, we have the native conformation that we form using electrostatic interactions and hydrogen bonds. So the native normal folded protein structure. And we have 4 different levels of structure. Our amino acid sequence is just our primary level of structure. Our secondary level of structure consists of our alpha helices, beta pleated sheets, and then we have our beta bends or beta turns. Under these beta bends and beta turns, we have a lot of hydrogen bonding that hold the alpha helices, beta pleated sheets, beta turns together. And then, of course, it's going to be determined based off of our primary structure, which is going to determine everything else. Our tertiary structure is the folding of a single polypeptide bond, and that's held together by covalent and non-covalent interaction. The one covalent or true type of bond that we have is our disulfide bridge, which is formed between cysteine molecules. And our non-covalent interactions were the hydrophobic, electrostatic interactions, and then our hydrogen bonds. And then our last level of protein structures are quaternary structure, which is not found in all proteins. It's just if they have multiple polypeptide strands that are wound together to make up the full enclosed protein, that's where we have a quaternary structure. So if we go back over to the left, we have several different types of protein. We have our fibrous protein, the one that contains a coiled coil. This is going to be our keratin, our supercoil. Other fibrous proteins are collagen. That's the most common one that we talked about. With our collagen, that's our triple helix. We have a couple different amino acids that are required. We have glycine, proline, which of course is an amino acid or an alpha helix breaker. And then I have lines going all the way up to our hydroxylysine and hydroxyproline. On this line going to hydroxylysine, I have it listed out that this is often glycosylated. That means that hydroxylysine often has a sugar added to it. That's that glycosyl. Tells us that there's a sugar added to that as well. Those are all post-translational modifications that happen. In order for synthesis of collagen to happen, we need a coenzyme and we need a couple cofactors. The coenzyme is going to be your vitamin necessary for it to work, so that's going to be your vitamin C. And then your minerals required for it to work is going to be your iron and copper. We need iron along with vitamin C in the same step, and then we need copper for cross-linking, lysyl oxidase. A deficiency in collagen, or deficiency in vitamin C in particular, you end up with scurvy. And then our collagen's able to be degraded using special enzymes that are called matrix metalloproteinases, or MMPs. These MMPs are really good for degrading extracellular matrix, which is where collagen's usually going to be found. Lastly, we have our globular proteins. With our globular proteins, these are highly folded. And the interior of these really folded proteins contain a lot of nonpolar amino acids. Remember, nonpolar are going to stay away from our polar matrix that we're transporting it in. Patterns of these highly folded globular proteins are called motifs. And then we have the stable patterns, because motifs, you pluck them out and they just unravel. Stable patterns are called domains, where you pluck those out and the pattern remains the same. We can denature these globular proteins using heat. Remember, scrambling eggs. We can use extreme pHs like our gastric juice, both basic and acidic work, or we can do mechanical shearing, which is where we talked about making angel food cake. If you have defects in folding, there's a couple different things we can do. We can either degrade it or we can accumulate it. If we degrade it, that's what happens with cystic fibrosis. We make a protein that's not folded correctly and the body degrades it right away. Versus things like sickle cell anemia, where it's folded incorrectly, but the body accumulates it because there's a reason for it. When we fold these globular proteins, it's going to decrease entropy because there's a very specific thing that we're going to do. So we're actually decreasing that entropy, that S. So we're making the cell more organized, which is what we want to do. The intermolecular forces, especially your electrostatic interactions, are going to be really important for your globular folding. And then we need chaperone proteins to help in the folding process. And then the primary sequence, or the primary structure of the amino acid listing is going to be extremely important for helping us fold correctly. If you have one little mutation in that amino acid sequence, that protein can't fold correctly. Okay, so that completes our first concept map. Thank you very much.